Hello everyone and welcome to the first International Electronic Conference on Toxins. This conference has been organized by MDPI and the Toxin Journal, and I would like to thank our chairs and conference committee for making this conference possible and all the participants for joining us today. The conference is now open for discussion, so until January the 31st, you can read and comment all submissions presented to the conference. 3 other live sessions are scheduled for tomorrow and the next week, so please check the webinar's website for more information and to register for free. Our session today and the one of tomorrow focuses on mycotoxins and their involvement in fungal plant infection, animals-humans toxicity, and potential therapeutic applications. Our chairs today are Dr. Marc Maresca from the Institut de Sciences Moléculaires de Marseille, Marseille Université in France, and Dr. Isabelle Oswald, Research Center in Food Toxicology in France. We will have 5 speakers today, and after each presentation, we will hold a Q&A session in which we will address the questions submitted by live online audience. So if you're joining us live, please submit your questions at any time during the webinar in the Q&A box. And as we have more than one speaker today, please mention the name of the speaker together with your question. I would like to remind everyone that this webinar is being recorded and that the recording will be posted soon at the webinar's webpage. Now I'd like to introduce our session chairs. Dr. Marc Maresca is currently a researcher at the Marseille Université. He was awarded his PhD in biochemistry from the Université Paul Cézanne in France in 2003, where he worked on toxins causing intestinal pathologies, including mycotoxins. In 2003, he moved to England to work on the bacteria enteropathogenic E. coli in the laboratory of Brendan Kenny. He then moved back to France to continue his work on mycotoxins, mainly deoxynivalenol, studying its effects on human health. He's currently working at the Marseille Université, and his research aims, in addition to study mycotoxin effects on health, to identify and develop new molecules with antimicrobial properties, such as natural, synthetic, and bio-inspired, including the ones produced by MicroRNAs. Our second chair today is Dr. Isabelle Oswald. She's the head of the INRA Research Center in Food Toxicology in Toxalim in Toulouse, France, where she leads a research team of 20 people. Her team has 2 main goals: to characterize the production of mycotoxins and other secondary metabolites by fungal species, and to determine the toxic effects of mycotoxins in rodents and pigs using cellular, tissural, and animal models. Dr. Oswald's work currently focuses on the effect of emerging mycotoxins and toxin mixtures. She has more than 200 international publications and is Thomson Reuters Calibrate highly cited researcher since 2018. She's an expert for the European Food Safety Agency and for the Joint Food and Agriculture Organization of the United Nations and World Health Organization. In 2018, she received a Lifetime Achievement Award from INRAE, and this January, she was appointed Knight of the French Legion of Honor. So now I would like to hand over the floor to our chairs, and to all of you, enjoy your webinar.
Thanks a lot, Elena. I'm trying to, yes. Okay. Hi, I'm Marc Maresca from Marseille University. I would like to thank Professor J. Fox, as well as Elena, Bonnie, and Charlotte, and all the staff for making this webinar possible. I would like also to thank the chair, the speakers, and all the participants to this webinar. So the webinar will be on mycotoxin and their role in plant infection, their toxicity for humans and animals, as well as their potential use in medicine. So what about mycotoxins? Mycotoxins are secondary metabolites produced by fungi, and these molecules are present in food. And when we look at them as structural chemistry, you can see that they belong to different family of molecules that will be more exposed during the webinar. And mycotoxins are also diverse in terms of action and effect, as some of them are involved in the effect of the fungi on plant immunity and cell death. Other mycotoxins can play a role in the ecology of the fungi with other microorganisms of the soil, like bacteria and fungi. And finally, mycotoxin can be toxic for humans and animals, as well as a potential use of them for drugs in medicine. Here are the program of the 2 days of session of the webinar, and I will now let Isabelle Oswald introduce the first speaker of the webinar. Thanks a lot.
Thanks, Marc, and thanks to all of you for being here. The first speaker today is Professor Fiona Doohan. She is from University College of Dublin in Ireland. She had a Bachelor of Science in this university, and then she did a PhD in John Innes Centre in UK. After a postdoctoral position over there, she moved back to Dublin, where she joined the faculty as a plant pathologist. Her recent work is focused on understanding the role of mycotoxin in plant-pathogen interaction and developing environmentally sustainable means of increasing crop sustainability. She's also the co-founder of the company eSeed that focus on developing biologicals for the enhancement of crop productivity. She's a member of the scientific board of the Global Wheat Initiative, a G20 initiative that promotes wheat research at international level in both developed and developing countries. Fiona, the floor is yours.
Thank you very much, Isabella. So today I'm going to talk about our work over the years on looking at how plants respond to mycotoxins. And the mycotoxins we're particularly interested in are the trichothecene mycotoxins produced by Fusarium fungi, and in particular deoxynivalenol. So we work a lot on wheat, and as most of you know, it's one of the top 3 sources of human calories, and we have a huge pressure to increase wheat production in order to feed the global community by 2050. And we lose about 25% of the grain or the yield from wheat because of plant diseases. And one of the most important diseases is Fusarium head blight disease, which, as you see here, attacks wheat heads and causes premature bleaching. And inside, of course, you get the mycotoxin contamination of the grain, which obviously, as most people here know, has huge health consequences. It's a very difficult disease to control. Fungicide, the use of chemicals, is inconsistent at best, and breeding crops or varieties of crops that are more resistant to the disease is considered the most environmentally sustainable strategy, particularly now in the EU as we move towards the Green Deal goals and targets. So we work on Fusarium resistance to wheat resistance to Fusarium, and we particularly focus on the effect of Doohan on wheat. So Doohan, deoxynivalenol, is a trichothecene mycotoxin. It's obviously toxic, but it's also toxic to humans and animals, but it's also toxic to plants, and it facilitates the development of disease. So we call it a disease virulence factor. It causes cell death in plants. It alters membrane structure. Its classic function, of course, is the inhibition of protein synthesis. And when you apply the pure toxin to wheat heads, it causes the same symptoms as does the disease-causing organism. So when you put Doohan onto wheat heads, you get disease. And it affects all sorts of different processes within plants, including cell death, and the effects on cell death are very dose dependent. Low levels, we've shown, can inhibit cell death, and high levels promote cell death. So just to show you what I mean when I say that Doohan is a virulence factor, here's 2 different varieties of wheat. One is Doohan resistant. The red arrow indicates where we inject Doohan into the plant. There is no visual effect here on the left. If you look at the Doohan susceptible variety on the right, you see you have premature bleaching of spikelets. The same symptoms that we get with Fusarium when we inject Fusarium into spikelets. So we are looking to enhance wheat resistance to Fusarium and to the mycotoxins it produces. We look for genes that enhance resistance or that enhance susceptibility, because we can manipulate those genes through classic breeding or through GM approaches to enhance disease resistance in crops. So over the last 15 years, we've used various techniques to identify wheat genes that are responsive to the mycotoxin deoxynivalenol and those that enhance resistance to the mycotoxin deoxynivalenol. And we did that by sort of association studies where we screened populations segregating in terms of their Doohan resistance and identified which genes do they differ in, particularly at the functional level, at the RNA level, which genes are turned on in resistant varieties and not turned on or turned on to a lesser extent in susceptible varieties. And then we go on to prove whether or not they actually directly affect Doohan resistance. And we've done that for a whole range of genes. And today, the title of the talk is about the evolutionary story. I want to tell you a very nice story. Well, I think it's very nice, of course, I'm biased, about the relationship between Fusarium and wheat and the role of evolution in the relationship between wheat and the mycotoxin Doohan. So I want to tell you about this gene. We start off with this gene, and this gene leads to a story on different plant genes. This gene is called TA frog, Trichomesticum wheat Fusarium resistant orphan gene. So through these studies, we identified this gene as being responsive to Doohan. Evolutionary wise, it's a very interesting gene. You only find it within a small subclade of the grass family in plants. We study it in wheat, you get homologs or matching genes in other Poideae, but you don't get it in the closer relatives outside the Poideae family, such as bamboo or rice. So it's an orphan gene. It's a gene that is very young in terms of evolution. It's a rapidly evolving gene, or a recent evolution. And we showed that this gene is turned on in plants in response to Doohan, in response to Fusarium that produces Doohan, but not when you take away the Doohan production from the fungus. So look at this graph. Let's take it slowly. All this graph is showing you is wheat heads where we have treated with either the wild type fungus in black columns or the same fungus where the Tri5 gene, the main gene responsible for Doohan production, has been deleted. Okay, so there's no Doohan production in this strain here. So what we showed here in the graph is the relative gene expression. So the higher the bar, the more TA frog gene expression. And we analyzed that up to 5 days post-inoculation with Fusarium. So this is wheat spikelets inoculated with the FHB fungus, Fusarium graminearum. And you see that the gene expression peaks at 2 days post-inoculation. It's turned on in response to this fungus at this point. We don't see any gray bars, or they're very low. That means the gene is not turned on in fungus that is producing it's producing disease symptoms. We also measured the level of disease in the measure of fungal biomass. And the gray bars, the mutant fungus caused about half of the level of disease that the wild type fungus produced, but we didn't get half the level of TA frog gene expression. So this gene was very responsive to the ability of Fusarium to produce Doohan. And all the dotted line is showing you there is we concurrently measured Tri5 gene expression, which you're seeing here, in the fungus. And we saw that the peak in TA frog expression in wheat corresponded to the peak in Tri5 gene expression in the fungus. We overexpressed this gene in wheat. We put an extra copy into wheat. We had 2 overexpression lines. They're there in the bottom, overexpression line 1 and 2. And we compared their resistance to Fusarium head blight compared to the wild type plant. And we saw that when we put an extra copy of the gene into wheat, we had an increase in disease resistance of about 25%. So then we could categorically say that TA frog contributes to FHB resistance and to Doohan resistance. We also showed it for Doohan in wheat. So what's this little interesting protein? By its nature, there's no motifs in this protein, no characterized motifs. It's rapidly evolving. It's nuclear. That's all this is showing you. We showed that it was a nuclear protein. This is nuclear staining in blue, a yellow fluorescent tag added on to the TA frog, and they both co-localize. So it's a nuclear protein. And we also did yeast to hybrid studies with a wheat library to determine what proteins in wheat might this little, what we call orphan protein, interact with. And we identified a whole range of proteins. And the upshot of it is that based on the identity of the proteins, it points to a role for TA frog in the reprogramming of metabolism and transcription in general in response to Doohan. And I want to tell you about one particular gene, SNRK. Okay, so SNRK as an AMP kinase, homolog in plants, ortholog in plants. It's a key role in sugar signaling. It's involved in many important processes in eukaryotes. And this just shows you that it's also involved in defense against pathogens. So people who work in the mammalian world would know the ortholog of this is AMP kinase. So a very interesting little gene involved in sugar signaling and regulation of metabolism. It's been shown in plants to be involved in defense against viruses, which you see in A. And in B and C here, the authors showed that it was played a role in defense against bacteria. So that was one of the proteins that we identified as an interactor with frog. And the next few slides are going to show you 5 points. Our SNRK, we showed that there's a lot of different copies of the SNRK gene in wheat. We showed that the one we found is functional and that it does indeed, we validated that it interacts with frog. We also showed that its activity, the protein activity and the kinase activity, I should say, are induced in response to Doohan. We showed that the SNRK itself contributes to plant resistance to Doohan. It enhances the activity of frog, enhances the activity of the kinase when it's overexpressed in plants. And at the end then I'll show you how a very nice study by other researchers recently showed that frog competes with a fungal effector to interact with SNRK. So first of all, our SNRK's functional. We threw it into a yeast mutant lacking its own SNRK, which is SNF1, and we restored activity and growth. So brilliant. We were sure then there was a functional SNRK. We showed that the one that we work with is specific to a particular subfamily of SNRKs in plants. All the second part here shows you is that when you cut the yellow fluorescent protein in 2 parts, you stick one part onto frog and the other part onto SNRK here in red. The two come together, you get yellow fluorescence. So that means there's an interaction. You see the yellow, the blue here is the nucleus. You see the same thing actually happens with frog itself. It homodimerizes. But frog doesn't interact with every SNRK that is in the plant kingdom. The interaction is specific to a particular subfamily. And that's what it was showing you in the last one there. So another SNRK, for example, SNRK 2.8, no interaction. And then we showed that while Doohan treatment had no effect or downregulated to some extent the transcription of SNRKs, TA, triticum vesticum SNRKs, wheat SNRKs, it increased both the kinase activity and the level of active SNRK. So when we treated with Doohan, so that's what you're looking at. If you just look at A as an example here, we're measuring kinase activity up to 3 days post-inoculation of heads with either mock treatment in light gray or Doohan in dark gray. And you see that over time, you get an increase in both treatments, and the increase is significantly higher with Doohan as compared to mock. And we saw a similar thing then when we looked at the actual kinase or PAMK relative activity. Okay, we use a technique in our lab called VIGS, or virus-induced gene silencing, to knock down the expression of genes. And we did this for SNRK using 2 different constructs. We do 2 independent constructs for gene silencing. They're called BSMV S1 and S2. And what we found that when you silenced or you reduced the transcription of SNRK in S1 and S2 here, you get more Doohan-induced bleaching than you do in plants where we just treated with the empty virus, where we weren't silencing SNRK. So that showed us that SNRK contributes to Doohan resistance. And I guess what was really nice recently for us was a validation of that work, where a really nice study showed that, yes, they validated our findings that SNRK interacts with frog, which is really nice. And they did some really nice work to identify a fungal effector. So when fungi attack plants, they produce effectors that go inside the cell. Here you have a cytoplasmic effector, OSP24, to induce a host response. OSP24 effector enhances cell death, at least in transient expression systems. And they showed that this fungal effector also interacts with SNRC. And they postulated that OSP24 competes with TA frog to bind SNRC. When OSP24, binds to SNRC, it destabilizes it and towards the ubiquitination system. So they postulated that the TA frog SNRC interaction stabilizes SNRC and enhances resistance to FHB. So that was really nice validation of the work and really nice finding here of a new fungal effector competing with plant gene TA frog to bind SNRC. And what's really interesting from the point of evolution is that both of these are orphan genes. TA frog is a new gene in wheat. OSP24 is a new gene in fusarium. They are both recently evolved genes, which is really, really nice. I said earlier on that we did a yeast to hybrid study of a Doohan-induced fusarium library, and we identified SNRC, but we also identified other proteins that interact with TA frog. And I don't have huge time, I guess, to go into all of those today. I'll just mention that one of them is a homeodomain protein, which is potentially involved in chromatin remodeling. There is another one, which actually told B defense-like protein, which links to autophagy and to a family that have classically involved in abiotic and biotic defense responses. And I would like to mention and point you to a publication, because we don't have time to go through all the results, on a NAC transcription factor. So transcription factors, as you know, regulate gene activity. NAC transcription factors are a family of transcription factors that are specific to plants. They're a very large family of transcription factors. They have many diverse functions in plants. Many are involved in abiotic stress resistance. Some are involved in biotic stress resistance. And here we had shown that TA frog interacts with one such NAC. And again, this is a story of evolution because it's not a typical NAC family. This diagram shows you the subdomains you find A, B, C, D, and E in a typical NAC protein. This is typical. Our NAC is atypical. There's missing the typical C domain, and it has what we call a TA-NAC LD1 specific subdomain. So TA-NAC-like gene, as L stands for like, because it's not a typical NAC subdomain architecture. So this gene has an extra domain in here. I don't have time to tell you about this today, but I give you the publication below. If you looked at the graphs in terms of the effect of Doohan on the expression of this gene, you would think that you were looking at the same graphs for SNRC. It's responsive to Doohan. It's responsive to Doohan production by fusarium, only responding to fusarium when it's producing Doohan. And we overexpressed this gene in wheat, and we showed through overexpression, this gene enhances FHB and Doohan resistance in wheat. And we published that over a year and a half ago now. So really interesting little gene, another transcription factor, and we're now doing a lot of in-depth studies on that. So I come back to the story of evolution. So plants have evolved defense mechanisms against Doohan. The mechanisms are often not general fusarium defense mechanisms. They are specific to the production of the toxin by fusarium. And indeed, one of the areas that we're very interested in now in the plant world and looking at is what is the effect of lower levels of Doohan on the plant transcriptome. Very often we've been looking at these levels that are physiologically not what the plant is faced with in the first few hours of disease. And we think that actually the role of the toxin is very different depending on the concentration. And so now we begin a lot of studies looking at the effect of very low levels of Doohan on the plant system and on plants' defenses. What we have showed through frog, even through the clade and the particular subfamily of SNRCs that interact with frog, the particular subfamily of gnats that interact with frog, is that the family of Poidia, the family of wheat's closest relatives, have evolved taxon-specific mechanisms to ameliorate the deleterious effects of Doohan. So you find these genes in all the hosts of Fusarium. So you can delineate, the closer they are to the host, so wheat, barley, oats, etc., you find homologs of the gene. And then as you move out to other species, you lose them. Of course, it depends on the mechanism of infection, etc. And we've shown that these genes play a direct role, not just in the Doohan response, but enhancing disease resistance. So by studying the response of plants to Doohan, you can better understand the plant toxin interaction, the plant fungus interaction, and you can use that information to enhance FHB resistance. And now we're working with industry to look at the role of certain genes and allelic variants of these genes in disease resistance in wheat, but also potentially in other crops that don't host these genes. For example, what would happen if we put TA frog into maize and looked at ear rot? Would it affect ear rot? TA frog is not in maize naturally. So we've shown that SNRC may help to protect plants against Doohan, maybe by reducing ROS levels via autophagy. That's something that the authors of the effector paper also speculated. And they've speculated that programmed cell death is another potential mechanism whereby SNRC might contribute to Doohan tolerance. And we've actually shown as well that TA frog can directly affect hormone-induced cell death in plant systems. Finally, one of the things we're doing at the moment is we're looking at the effect of frog, but also the NAC gene on the plant transcriptome. What's the global reprogramming of the wheat transcriptome in response to these genes? In particular, we're interested in finding out what genes does the NAC transcription factor regulate. So I'll conclude there. That's just from an overview from the plant side, looking at how plants respond to Doohan, what we can learn from that. I'd like to thank the people in the lab, the collaborators who worked on that work, and the funders. Thank you very much.
Thanks a lot, Fiona. We don't have question yet, so while people are typing, I have one question. You almost answer it because you say that the gene hasn't any homologue in maize. I'm working on vertebrates, on mammals, and the question is there an homologue in mammals?
No. So we work a lot on orphan genes, not just for Fusarium, for lots of diseases in plants. And our screening process, we ask, what's the evolutionary story here? So what are the plants that, so obviously ear rot is a different mode of infection in maize. So we stick to the small grain cereals like wheat, oat, and barley. What are the genes you only find in those that respond to Fusarium? We work in another disease that affects only wheat. So we ask, what genes are unique to wheat that you only find in wheat that respond to that disease? So by our selection mechanism, immediately we rule out vertebrate genes. In saying that, we're always looking at very simplistic systems. We're not looking at quaternary structures of that, but basically it's a small disordered protein. And I think it's a chaperone. It is a nuclear signal. It is a nuclear export signal. It moves things around the cell and brings things together in response to the toxin.
Okay. While I don't see a question.
I have some, Isabelle.
Okay.
So thanks a lot, Fiona. So may I ask you if you have ever looked at a link between your gene and enzyme known to detoxify Doohan? Because it's already known that by introducing some glycoside transferase in wheat or other plant cells or plants, you can increase the resistance to Doohan and Fusarium. So is it a connection between the two? Or?
So not directly between the glucosyl transferases that were published or the homologs. We did find glucosyl transferases in our very original studies that were co-regulated with this gene, but they're very different potentially in terms of their function. What we're doing now is looking at the effect of TA frog overexpression on the whole transcriptome. And actually, we're just looking at that data and mining it. We're not seeing any major effect on those glucosyl transferases, though, it has to be said. I think that the role of our gene, ultimately, I might be wrong, but I think that it is really in reprogramming the cell system and stabilizing, and particularly probably towards cell death, but in a very refined way that we haven't actually got the correct tools yet to analyze, I would say. But certainly, it was one of the first things that we looked for. We also have cytochrome P450s but they are not the classic cytochrome P450s. I think the genes were the UGTs and the cytochrome P450s that we've found probably play a role in hormone balance in the cell and regulating, for example, IBA versus IAA, indole acetic acid versus a store form indole butyric acid, and other hormones in the cell, rather than there's no phylogenetically, there's no link with the published UGT. Yeah.
Thanks.
Okay, maybe we are still on time, so I have another question. Fiona, how specific is it to Doohan? So for other trichoticin, and how?
So we haven't tested for other trichothecenes in our thing yet. We did plan to do that, and then we didn't do it, to be honest. What we do know for all the transcriptome database, in terms of other treatments, it's very specific to toxin. Okay. So developmentally and others, we haven't tested. What we did test is cyclohexamide at one stage. And we found that there was a response to cyclohexamide. And I'm trying to remember, I hope I remember correctly when I'm saying that the effect was nowhere near as great. Okay. That it was a time shift. It's not that the magnitude might have been as great, but that there was a huge time shift in terms of molar equivalent responses was much later.
Okay. Okay. So thank you, Fiona. And if you have questions, you can still put it on the question and answer part, and we might have time at the end of the seminar to answer other questions.
Cool. Thank you. Thanks
a lot, Fiona. So now we will listen to the second speaker of today, that is Philippe Pinton from INRA Toulouse, Toxalim. And Philippe is a PhD engineer working from now 20 years in INRA Toulouse, Toxalim, and is particularly working on mycotoxin at low dose and in mixture using cellular and animal models, mainly the pig, but also human cells. Now, Philippe, is up to you.
Hello, everyone. So first of all, thanks to the organizers and the chairs of this webinar for inviting me to present these results. Our study was focused on the co-occurrence of Doohan and emerging mycotoxin in pig feed and on the assessment of their combined toxicity on intestinal cells. I will start by background information, first about regulated mycotoxins. All of you know that more than 50% of cereals and derived products are contaminated by mycotoxins. Regulation and recommendation exist for the most known toxic and prevalent mycotoxin and are determined according to their individual toxicity. Among these mycotoxin, you know deoxynivalenol, Doohan, produced by different fusarium. It is toxic for the intestine and the immune system. On the other way, exist emerging mycotoxin that are recently discovered, poorly investigated, not regulated. They contaminated cereals, food, and feed commodities. They are co-occurring with regulated mycotoxin and mainly produced by Penicillium, Aspergillus, Fusarium, and Alternaria. Pig is an important species for mycotoxin because due to its diet rich in cereals, it can be exposed to high concentration of mycotoxins. And in addition, it's one of the most sensitive animal species to these food contaminants. The intestine is also important because the intestinal epithelium is the first barrier against food contaminants. It plays an important role in nutrient uptake, barrier function, or immunity. The objectives of our work were to determine the co-occurrence or the occurrence of emerging and regulated mycotoxin in pig feed, and then to assess on pig intestine the combined toxicity of emerging mycotoxin and Doohan on the basis of their realistic proportion in feed, and then to try to determine the nature of their potential interactions. The first part of this work was performed by our colleague from Biomin in Austria. They performed a worldwide survey between 2014 and 2018. They analyzed more than 1,000 samples from finished feed, coming from mainly Europe and North America. Among these samples, 524 were from pig finished feed. In this sample, they identified 235 different fungal metabolites. And among these finished feed, pigs feed, 88% were co-contaminated with Doohan and other fungal metabolites. And we focused on these co-contaminated samples. Here is the list of the main metabolites we have identified in these samples, the 25 more occurrent with a percentage of presence in the samples. They were mainly produced by Fusarium, Aspergillus, and Alternaria. If we have a look to their co-occurrence with Doohan, you can see in this column that the percentage of co-occurrence is high for all the metabolites. So we selected 10 emerging metabolites among the most occurrent that were commercially available, and we had apicity because it was already shown as a toxic compound. The first part of the study was to assess the individual toxicity of this emerging metabolite on pig intestinal cells. We chose the IPEC1 cells that were exposed 48 hours to the different metabolites. Cell viability was evaluated by the measure of ATP content, and we obtained the following results: the dose effect for the different compounds. We verified that for Doohan, we obtained toxicity in accordance with the previous published results. We identified 3 metabolites, brevianamide, cyclo-L-proline, L-tyrosine, and tryptopol, that were, in our condition, non-toxic. 3 metabolites were qualified as moderately toxic metabolites, rofusarin, emodin, and eniatin B1, with IC50 ranging from 14 to 19. And 4 metabolites, apicidine, eniatin A1, bovarecin, and eniatin B, were classified as highly toxic metabolites with IC50 ranging from 1.5 to 4.4 micromolar. For the study of the combined toxicity of these toxins with Doohan, we first tried to verify if there were a correlation between the concentration of Doohan and these metabolites. There was no correlation, despite the high percentage of co-contamination. So we imagined plausible mixture by using the data distribution and the concentration at the different quartiles. You have here an example of the distribution for the Doohan contaminated samples. And we use the values of the quartile P25, P75, and median to imagine mixture in which, for example, for the first scenario, we had a low concentration of emerging metabolite and high concentration of Doohan, or the reverse ratio with high concentration of metabolite and low concentration of Doohan, or the median concentration. Then we converted in micromolar the concentration that were expressed in microgram per kilogram of feed by assuming that 1 kilogram of feed is diluted in 1 liter of gastrointestinal fluid. We obtained the different concentration for all these metabolites that are indicated here. And from these values, we prepare mixtures as shown as an example here with Doohan and brevianamide. For example, for the combination of P25 brevianamide, P75 Doohan, we obtained a ratio of 1 to 21. For the ratio median, the ratio was 1 to 7. And for the ratio P75, P25, it was 1 to 2. So based on this way of calculation, we prepared the different dilution of mixtures that were tested for the cytotoxicity in parallel with the compound alone. Here are the results that we have obtained first with for the combination of Doohan and the non-toxic metabolites, And I will detail this graph because all the graphs will be presented in the same way. You have on the x-axis the concentration of emerging mycotoxin in red and of DON in blue. The red line and symbols are for the emerging mycotoxin, blue line and symbols are for DON, and the mixture is presented in black. For the statistic, the red star corresponding to the difference between emerging mycotoxin and mixture, and the blue hashtag correspond to the difference between DON and mixture. So here is the first result for the ratio P25, P75 for brevianamide and DON. And here the two other ratio. You can see that we have the same trend for the curves. Exactly the same result for cycloelproline, L-tyrosine, and DON, and for tripropol and DON. The main observation that we have when we observe this graph is that the combination gives the same toxicity or the same trend as DON alone. And this is the main conclusion, that the combined toxicity were similar to the one of DON for all the ratio. For the second group of metabolites, the moderately toxic metabolites, orofusarine, amodine, and eniatin B, we have observed for the ratio 1, for the ratio 1 on the left, that at this level of doses, which was quite low, the different metabolites were non-toxic. For the ratio 2, we observe toxicity for orofusarine and eniatin B. And you can see here that the combined toxicity of eniatin B1 and DON was lower than the one of DON. For the ratio 3, amodine was still not toxic at this level of doses. And the combined toxicity were in different conditions, but it was characteristic. They were lower than the one of DON, and this is also the main conclusion of this test, that the combined toxicity were similar or in certain conditions lower than the one of DON. And for the last group of metabolite, the highly toxic metabolite, eniatin B, bovericin, eniatin A1, and apicidine, we have observed that at this level of doses, these 4 compounds were not toxic for the ratio 1. For the ratio 2, all the emerging mycotoxin were toxic except bovericin at this level of doses. And the combined toxicity were lower than DON alone, except for bovericin. And for the ratio 3, all the compounds demonstrated toxicity because the doses were higher. And the combined toxicity were, depending on the compound, but lower than the one of DON. And this is also the main conclusion, with the combined toxicity of this metabolite with DON were similar or in certain conditions lower than the one of DON. So to conclude this work, in worldwide peak finish feed, we have observed that DON and emerging mycotoxin are present together, despite a low correlation between their concentrations. Based on their toxicity on pig intestinal cells, the 10 most occurrent metabolites were classified in this order. Apicidine and eniatin A1 were the 2 compounds more toxic, whereas brevianamide, cycloelproline, L-tyrosine, and tripropol were shown as non-toxic. In most of the cases, the toxicity of the mixture with DON was similar or lower to the toxicity of DON alone. It means that if these results are confirmed for other organs or in vivo, it's important for animal health because it means that the toxicity of DON is not exacerbated when this mycotoxin is present in mixture with the different metabolites that we have tested in this study. I want to thank my colleague from Biosynthesis and Toxicity of Mycotoxin team, a team which is led by Isabelle Oswald, and also our colleague from Biomin in Austria for the analysis of the samples. And I thank you for your attention.
Thanks a lot, Philippe, for this nice presentation. I don't see any question for the moment, so I will start myself, and I may give the microphone to someone else next. It's nice. I knew about the data anyway. And I would like to know if you have ever tried yet to look at other parameters than cell viability. So something more like more subtle than the viability of the cell, could be like TR or inflammation or any kind of parameter other than the cell life or death statue?
Some results exist in the literature for some of these compounds about the TEER, the electrical resistance on this type of intestinal cells. More or less, the trend is similar. It means that the compound that we have observed with no toxicity have no effect on the TEER, and the highly toxic compounds show an important effect of the electrical resistance. From our point of view, we are starting a more detailed study on apicidine and other compounds which are interesting because we are starting a global overview of the gene expression of this compound on the intestinal cells when they are exposed to this compound. But we have not enough results right now.
Thanks, Philippe. Sorry. We have a question from Siska Grubel. So maybe, Siska, you can ask it orally if you want, if Elena can give you the microphone to be able to answer directly, or I will just ask.
Yes, thank you, Philippe, for the nice presentation. I'm just wondering, for mycotoxins in general, having a similar mode of action, for instance, within the trichotillins group, when they are co-contaminating feed, do you expect synergistic effects by definition, or?
What we have already shown in a cellular model is that when we combine different trichotillins, for example, Doohan or nivalenol or acetylated derivatives from Doohan, we observe a certain level of synergy at very low doses. Mm-hmm. And this is a really interesting result because the type of interaction change when the doses are higher. We reach additivity or antagonism, but really we observe a synergy at low level of doses. Mm-hmm. Right now, we have not explained the mechanism that can lead to this synergy, but this is what we have observed right now.
Yeah. Okay, thanks. Really interesting to see this phenomenon and then to find possible explanations. Okay, thanks.
You're welcome.
Good luck.
Thank you. Bye-bye.
Thanks, Siska, for the question. Thanks, Philippe, for the presentation and the answer. And if we want to keep on time, I will now give the microphone to Isabelle for the next speaker.
Thank you, Marc. The next speaker is Veronika Nagl. Veronika works as a scientist at the Biomin Research Center in Toul, Austria. She studies veterinary medicine at the University of Veterinary Medicine in Vienna. Thereafter, she did her PhD in the Christian Doppler Laboratory for Mycotoxin Metabolism at the University of Natural Resources and Life Science in Vienna, and her research was focused on the development of an healthy MSMS-based biomarker method to elucidate the metabolism of mass mycotoxin in rats and pigs. After a postdoc in the same lab, she joined Biomin Research Center in Thun, Austria in 2014. There, her main research topic is the investigation of molecular effect of Fusarium mycotoxin in livestock species. Special emphasis is put on employing omics technology and the utilization of result of biomarker development. So far, Veronika has authored and co-authored more than 20 articles, book, chapter, and patent, and presented her work in multiple international conferences. Veronika, the floor is yours.
Thanks, Isabelle, and hi everybody also from my side. It's a great pleasure to be here in my fake apartment and to tell you a little bit about the journey on Zearalenone and MicroRNAs that we did during the last couple of years. So these MicroRNAs have moved into the spotlight of research, and this is due to the fact that they're involved in many cellular processes and also diseases. So they have been associated with diseases like different types of cancer, but also, for example, pulmonary hypertension. There also have been diagnostic kits based on microRNAs rolled out, again for cancer, for example, but also osteoporosis. And at a later stage, microRNAs have been discussed as therapeutics for important diseases such as multiple sclerosis, nowadays also corona, and maybe some less severe syndromes like hair regrowth. In case you're not too familiar with these microRNAs, here are some key facts. So they are small, approximately 22 nucleotides long, non-coding RNAs. The major mode of action or the major role is that they are negative regulators of gene expression at post-transcriptional level. You can see here on the right a figure, and you can see that these microRNAs, they undergo a certain maturation process. So these pre-microRNAs, they have this classical hairpin structure, but in the cytoplasm, they are then converted and they can do their action, which is repressing either translation of mRNA or directly degrading mRNA. These microRNAs have some super nice tools, or features, let's say it like this. So they are released from the tissue. And this means you can detect microRNAs in biological fluids such as serum, urine, saliva, and so on. And this enables us non or minimal invasive sampling. They're also pretty stable. So they have been tested, for example, for pH stability and also during multiple cycles of freezing and thawing. I mean, I wouldn't recommend that, but in general, the robustness has been tested. Overall, there are some non-expensive analyses for microRNAs available. Of course, it's always a discussion what is expensive, what is not, but some qPCRSs are pretty straightforward. And for some of these microRNAs, it has been described that they're tissue specific. So for example, they can really indicate liver toxicity or cardiotoxicity. I mentioned before that the research in this field kicked off, and this is since 2000. So there are two reasons for that. First, the first human microRNA has been identified in around 2000. And then later on, advances in technologies such as microarray or next generation sequencing really enabled more and more research in this regard. When we talk about research on mycotoxins and microRNA, I cannot say that research has exploded in that regard. So here on the right, you have the numbers of publication when you put these two keywords together. And please be aware that the scale is different on these two figures. When we started our research on Zearalenone and microRNAs, there were two publications out there. So colleagues from Poland investigated the effect of certain microRNAs on liver and on intestine in pigs. And another publication dealt with the specific role of microRNA 7 on follicle stimulating hormone synthesis in pigs. Why Zearalenone? I mean, I think you're all pretty familiar with this mycotoxin and that its major mode of action is estrogenicity. We were interested because when we talk about microRNAs and estrogen signaling, these compounds seem to have really a key role in this process. So a lot of research has been done on microRNAs and estrogen-related diseases such as breast cancer. So we thought, okay, due to the similarity on the mode of action, we have a good chance for Zearalenone as well. Also, we all know that pigs are most sensitive to this mycotoxin, but it's pretty hard to diagnose Zearalenone-induced mycotoxicosis on field. We have these biomarkers of exposure available that work pretty nicely under controlled condition, but it's really tricky to employ them on field. And we do not have any mechanism-based biomarkers available for diagnosis. So when we started our activities in this field, we had three major research hypotheses. First, we wanted to find out if dietary SAN affects the global intestinal microRNA expression. So we did not want to check just individual microRNAs, but we went for an untargeted approach. The same was for uterus. The reproductive tract is the major target of Zearalenone, so that's why we choose this organ. And then in case that we saw something in tissue, we wanted to follow up on serum and to check if this tissue response is indeed reflected in the circulating microRNA pool. To this end, we performed a proof of principle trial where weaned female piglets were exposed to different treatments. You can see here 4 groups. So we had a control group and we had 3 groups with CEN contaminated feed. Concentrations were increasing, so from 0.2 ppm up to 4.6 ppm. Concentrations were quite high, but as I mentioned, this was a first proof of principle trial. We've exposed the piglets for 4 weeks, and during this time period, we regularly checked on body weight and also measured the vulva size, for example. On day 27, we collected tissue from jejunum and from uterus. We then extracted the RNA from this tissue via a microRNAs in minikit. We determined RNA concentration, quality, did the library preparation and the sequencing with 10 million reads per sample. And then afterwards, with a customized bioinformatics pipeline, we did the data analysis. So reads were aligned to reference databases and mapped against Sussgroffer genome assembly, and at this point, the 10.2 version. And with this, let's jump into the results. So it's usually in bioinformatic analysis, we started with exploratory expression analysis, meaning we just wanted to have a global overview whether the dietary treatments had an overall effect on the microRNA expression in uterus and in jejunum. So here you have on the left the principal component analysis for microRNA expression in uterus, and on the right for jejunum. You can see on the left for uterus that microRNA expression in the control group in purple, and in the CEN low group in blue, that they form a cluster. And the same is basically for the CEN medium group in green and the CEN high group in red. So we have 2 distinct clusters, more or less, in this tissue. We cannot say the same for jejunum. So no clustering here. It more looks like a solar system, I would say. And this was basically also reflected then in the differential expression analysis. So there were no microRNAs significantly affected in any of the CEN treatment groups compared to the control in jejunum. I have to say we were pretty strict what we call significant. So we used adjusted p-value lower than 0.05, so adjusted for the false discovery rate. In contrast to jejunum, we found a couple of microRNAs affected in uterus. So in the CEN medium group, we had 16, and in the CEN high group, 67 microRNAs affected compared to the control. Most of them were upregulated, only a smaller proportion downregulated. The fold changes compared to the control were pretty pronounced, so up to 12.6 fold. You might also have seen that for the CEN low, there are zeros again. So we had no microRNAs differentially expressed between the control and the CEN low. And this was actually in line with our clinical observation. So in this figure here, we plotted the vulva area against the trial duration. And what you can see here in purple for the control and the CEN low group in blue is that the vulva area did not change over the course of the experiment. This is different for the CEN medium and the CEN high groups, where the vulva area increased significantly. So having a closer look on the results for the CEN high and CEN medium group, we wanted to know, of course, these microRNAs that affected in both groups. And we had 14 microRNAs that were changed both CEN high and CEN medium. You have a list of these 14 microRNAs here. Checking the literature, there have been different modes of action attributed to these microRNAs, also related to muscle development, for example. But we were specifically interested in the highlighted microRNAs because they all belong to this microRNA 503 cluster. So a cluster describes a set of 2 or more microRNAs that are described from physically close by microRNA genes. And we were specifically interested in that one because 503 has been postulated as master regulator of estrogen response in vitro. In this study, the authors exposed the MCF7 cells to estradiol, and what they really nicely did is they combined mRNA transcriptomics and microRNA sequencing to come to that conclusion. So in the next step, we checked these 14 microRNAs a bit closer, because we wanted to see, do we see the same response in the different groups? So are they upregulated or downregulated in both groups? And is there a certain dose dependency? Here you have a few examples on the microRNAs. So the normalized read counts for the individual microRNAs for the individual groups. And you can see that especially for the upregulated microRNAs, we had a very nice dose dependency. Not so much for the downregulated, I have to say. So sometimes they were downregulated always in both groups, but not in a dose-dependent way. What you can also see on this figure is that we had some individual differences between the animals. So this is quite usual in vivo experiments, but for some of these microRNAs, it was more evident than for others. And what I also wanted to mention here, we focused a lot on sequencing, of course, but we confirmed these results also by qPCR. So we selected certain microRNAs and we really checked for their expression via qPCR to get more trust in the results and to confirm we are 2 different quantification methods. So up to that point, we have to say, nope, in our study, there was no effect on the microRNA expression in jejunum, but definitely so in uterus. So the next step for us was to check for serum. Do we see the changes in uterus? Do we also see them in serum? For this kind of analysis, we switched to a targeted approach. So we selected, based on our results, 44 microRNAs and analyzed them via qPCR in the samples collected on the last day of the experiment. At first, we were a bit disappointed because we had a lot of hope in our microRNA 503 cluster, but the changes of this cluster were not well reflected in serum. You have here on this figure, we tried to compare the results from uterus and serum. So you have the fold regulation compared to the control group. All the green bars belong to the 10 medium group, all the red ones to the 10 high group. And the filled bars represent results from uterus, the dashed ones from serum. So basically what you can see here is that the high expression or the high fold change in uterus was not reflected in serum. For some of these microRNAs, like 450B, 5B, it was even the opposite. So we saw upregulation in uterus, but downregulation in serum. The only microRNA which behaved a bit nicely, I have to say, is this one, so the microRNA 542 3P. There are different reasons why sometimes the tissue alterations are not reflected in serum. It also has to do something with the specific release of these microRNAs to blood, and also that in the bloodstream, of course, microRNAs from different sources come together. But as I mentioned, we checked more than these microRNAs from this cluster. So the next question was for us, okay, are there any microRNAs that are good discriminators between the non-exposed and exposed piglets? Or to put it in other words, by measuring certain microRNAs, can we say that this animal has been exposed to zearalenone or not? And to do so, we employed receiver operating characteristic curve analysis, or in short, ROC curve analysis. So this type of analysis dates back to World War II, because back then it was pretty important to assess how good the individual radar operators are in distinguishing that a certain signal on the radar is a foreign plane, potential bomb attack, or just a noise. And this is nowadays used basically to tell how good a prediction model or a classifier is in discriminating between true positives and true negatives, or you can say between truly infected, non-infected, exposed, non-exposed. What usually the output of such a ROC curve is a curve and an area under the curve. So when you have 100% area under the curve, when you plot the true true positive rate versus the false positive rate, it tells you that with 100% power, 100% certainty, you can discriminate the negatives from the positive ones. Usually, ROC curves with area under the curves more than 75% are regarded as clinically very good and very useful. If you have ROC curves looking like this one on the right, the red one, which you have area under the curve of 50%, you should really get rid of your model or your classifier, and you should just take a coin and flip it around, because you will get the same result basically. You can just identify the true positives and the false positives by chance. So this is how it looked in our case. We employed this ROC curve analysis. What we also did is we built ratios. So we built ratios of up and down regulated microRNAs to further increase the sensitivity. And here are just two examples of this kind of ratios where we got pretty nice ROC curves. So over 85% for the ratio 455 5p to 493 3p, and also certain other microRNAs where we had very nice classification. Honestly, I haven't seen this kind of ROC curve analysis too much in mycotoxin research, but I really think for certain kind of questions, it's a nice tool that can be employed to get a bit more idea how good your potential biomarkers are. So for the last initial question that we had on the circulating microRNAs, we had to say, yeah, well, partly. But as usual in research, first results just create more questions. We asked ourselves, okay, we see changes, but are they indeed specific for Zearalenone? The next one is, are our results reproducible? And are these 1.5 ppm, so the Zeng medium group, is this really the limit to see something? And last but not least, we also, of course, wanted to apply our findings. I cannot promise you that we answered all these questions completely. I just want to focus in my second part of my talk just on some activities that we did to address these questions. So for the first one, on how specific the reaction is, we used an in vitro assay with MCF7 cells. So we exposed the human breast cancer cells to estradiol, zearalenone, and genistein. Genistein is a phytoestrogen, and we chose it as a model substance to test because it usually or often occurs in feed. So we thought it's pretty nice to compare it with zearalenone. We applied different concentrations and we measured then certain microRNAs via qPCR at different time points. On the right on the figure, you see as an example the results we obtained for microRNA 503 after 24 hours. So estradiol and Zen treatment caused a significant upregulation of this microRNA, whereas for genistein, we more or less saw no change. So based on these data, we could say, yeah, well, for this microRNA at this time point, it seems that the changes are specific for zearalenone, also because genistein is maybe more going to the estrogen receptor beta. But I have to add that overall, in this assay, we did not observe any clear time or dose-dependent effects. So the expression of microRNAs was pretty low, and also the fold changes of the positive control of zearalenone were lower than expected. So it's always a bit tricky in research. I mean, I guess you all know that, to come from in vitro to in vivo or backwards, or really to get the same results. And in case of microRNAs, there were also others who observed that. So there's a very nice study focusing on kidney toxicity based on oxatoxin and on microRNAs. And it was done in rats, and also here in vitro and in vivo results did not perfectly fit together. concerning the reproducibility of our in vivo results, we wanted to confirm them. Why? Of course, confirmation is also always important, but there is also a lot of money going in research into irreproducible results. So this figure here, I stole from Friedman, and it's just an example stating that approximately 50% of all the spendings in US annual preclinical research is done basically on irreproducible results. So what did we do? We tried more or less to repeat our first trial. We changed a few things. So we left out the highest SEN group. The SEN medium, we tried again to reach around 1.5 ppm in feed. So we achieved that just a tiny bit more. And the concentration for SEN low, we increased a bit to the 0.6. The sex was the same, so we used females again. Again, weaned piglets, trial duration was the same. What we also changed was the trial location. So this time we performed the trial at the VetMed Uni in Vienna in collaboration with the clinics of swine. And we focused on uterus and again employed small RNA sequencing. And we have to say we saw nothing for the 0.6 dose, SEN dose. So there we had no changes on the uterine microRNA expression. However, for the 1.5 concentration, we're pretty happy on the results. Here again, you have the one in dark blue are from the proof of principle trial, from the first trial I've presented. For the individual microRNAs, the fold change compared to the control. And here in lighter blue, you have the results for the repetition trial. So in general, all of the important microRNAs from the first trial that were upregulated were also upregulated in the second trial, and the same for the downregulated ones. For some of the microRNAs, we had a bit of a lower response, so lower fold change, but you can see for others, we pretty much yielded the exact same results. So we were pretty happy, at least on that, and that's why we also wanted finally to apply our findings. And we did so by checking on the estrogenicity of some zearalenone metabolites, namely hydrolyzed zearalenone, so HSEN, and decarboxylated hydrolyzed zearalenone, DHSEN. You can see their structures here, and they derive from enzymatic cleavage of zearalenone. So the enzyme opens the lactone ring. We used the in vivo model in pigs, and we exposed the pigs to a high SEN concentration and equimolar concentration of HSEN and DHSEN, and then checked for uterine microRNA expression. And in contrast to SEN, we had no effect of DHSEN and HSEN on the microRNAs. In correspondence to that, we also had no effect on the vulva area of these two metabolites, which nicely demonstrates that both HSEN and DHSEN are not estrogenic in this model. So to sum up a bit, or to conclude on our roller coaster ride. So yeah, zearalenone definitely affects microRNA expression. It is a toxicity marker, but as I've shown you, it really depends on the tissue, it depends on the dose and also the model. The small RNA sequencing or the omics in general, they of course a super nice tool to get a global overview to check on the global expression. But we really recommend to combine them with targeted approaches to confirm your results by two different quantification methods. There are a lot of influencing factors in vivo. This is nothing new. You can control for a lot of them, like breed, like sex, but the individual differences, they are there. So reproducibility is definitely something pretty important and maybe something a bit neglected. And if you want to go more straight to biomarker research, nowadays it's also, sequencing of serum or target fluids is nowadays easier, or let's put it like this, there have been some technological advances where the sequencing of these fluids is easier by now. So if you're not too much interested in where the microRNAs are coming from, the mode of action or this kind of thing, I would recommend you to go directly for this kind of sequencing. And you can be reassured that our journey will go on as well in this direction. So with this, I want to thank a lot of people who were involved in all these studies. So the colleagues from the VetMed Uni Vienna, from our collaboration partner TamiRNA. So they are really the experts in microRNA analysis. And of course, a lot of colleagues from BioMR&D. And I thank you for your kind attention, and I'm looking forward to all your questions.
Thank you, Veronika. There's no question yet, so I have 2 questions. The first one is just, did you check the mRNA in the serum of the second experiment?
No, we did not. So after
we were too disappointed by the first one that you didn't want to check in the second one.
No, I mean, I have to say we had more groups in this experiment, so there were more interesting groups, but no. So it would have been the plan, honestly, to do that in case we see something for the 0.6. But since this is what was not the case, and yeah, I cannot 100% agree that microRNA analysis is not expensive. So, you know, you have to set focus. So no, we did not.
Another question is, you clearly see that your mRNA, the 5031, exists also in human, as you demonstrate in cell line, and as it is already demonstrated to be in line with estrogenic hazard in another veterinary animal species?
So in general, these microRNAs, they're well conserved. So you have a lot of homology between the sequences across mammals. I mean, you have to check, it's not the case for all of the microRNAs, but for most of them. So for sure, this microRNA has also been, a lot of research has been done in rodents, you know, and in pigs, unfortunately, there's pig research on microRNAs usually goes into breeding, muscle development, but not so much into the disease right now. For the microRNA 503, there's not too much out there in mammals.
Okay, Laura has a question, but Laura, I think you can open directly your mic, no?
Yes. Hi. Hi, Veronika, thank you for your talk. I was wondering, you mentioned in the beginning of your talk that microRNAs are released into blood. I guess that some microRNAs will remain in the tissue and some other will go into the blood. Did you consider, or is it possible at all, to measure microRNAs in the supernatant of cell culture? Or did you consider doing some kind of ex vivo exposure of tissues just to check that maybe your microRNAs that you later want to see in serum are leaked into this culture medium?
Yeah, I mean, this is a very good question. So microRNAs, when they're released from tissue, there are different pathways. So it can be via exosome, it can be via extracellular vesicles or connected to proteins. When a microRNA is released, this is pretty strictly regulated, but the mechanisms behind are not fully discovered yet. There's also the theory that these microRNAs act as hormones, more or less. So it definitely makes a difference if you check, for example, in our in vitro experiment, if you check for the cells or the supernatants. On purpose, we checked for the cells because we wanted to, in this case, check really for the tissue response like we did in uterus and not for the release like in serum. But definitely, it really depends. You can also do the sample preparation, just focus on one subgroup of microRNAs. That's why it's also the difference. There is so many publications out there, but sometimes they really get conflicting results just simply because they focus on a certain subgroup or these things have not been considered. So yeah, it's a very valid point. We tried to do it in that way, but this is for sure not complete.
Thank you.
Thanks a lot, Veronika. I have a few questions in the time left. I have 3 questions.
Okay.
I I ask all, and then you decide what you answer.
Okay, this is also
The
fine.
first one is about the fact that you don't see any effect on the gut and see effect on the uterine part. It makes sense because it's a tissue estrogenic sensitive. So my question was about, did you test other tissues that are sensitive to estrogenic stimulation? So testes for men, also breast. I mean, the pig breast. So I don't know if you call this breast for pig, but mammals.
I know what you mean.
And the brain that is sensitive to estrogenic stimulation. First question. Second question is more about the stature of the female pig that you use. Did you ever try to see if, depending of their stature, puber, not puber, pregnant, I don't know the name in English, but menopause, I mean, without any more cycle. To see
menopausal pigs.
Yeah. And a final one that is more about a technical point of view. I was wondering if, for the fact that you don't see any serum modification, what is the amount of microRNA that you have in the serum compared to what you have in a tissue? Meaning that if the tissue really is just like 1% of something that is in the total amount in the serum, of course you will never see anything.
Okay, I tried to do all three of them super briefly. So I cannot say anything about other piglet ages. We used prepubertal 4-week-old female piglets in both trials, so I cannot say anything more. For the serum, yes, you're right. So for example, for microRNA extraction from serum is pretty tricky. And the ones where we checked for qPCR, most of them have had a low expression. Yeah? But as I also, the research showed, for some of them we saw something. But in serum, in general, it's more tricky, and that's why also sequencing is a bit more tricky. And for the tissues, yeah, well, for jejunum, honestly, we didn't expect much. But as I mentioned before, the colleagues from Poland saw something, especially in colon, I have to add, and they went for qPCR. So it's always a bit different, you know, qPCR sequencing, which cutoff values do you do? And no, we focus just on these two, and because we, as I said, we did sequencing, you know. Of course, you can go, it would be, I would have 20 ideas what to go next, you know. So I agree. Also, and of course, an experiment with males would be super interesting, or with mixed sex, let's put it like this. But no, we, unfortunately, so far, we didn't have the chance to do so.
Thanks a lot.
Thank you, Veronika. I'm afraid that now we have to move on. So Marc, now it's you to introduce the next
Thanks,
speaker.
Veronika. So the next speaker will be some collaborator from Isabelle also. Just going, so here, so it's Viviane Maruo, that is from Brazil. Viviane is an associate professor in veterinary toxicology in University of Tocantins. I'm sorry if I spell it wrong. She obtained her veterinary medicine degree at the University of Sao Paulo in 1993, a master's degree in 1997, PhD in 2002, and a postdoc position in 2010 in the field of experimental and comparative pathology at the University of Sao Paulo. She complete a CNPQ funded postdoctoral fellowship in 2015 at the INRA Toxalim unit in Toulouse. She was in charge of the graduate program in tropical animal science of our university from 2010 to 2014. Currently, she has a CAPES-funded visiting professor fellowship at Toxalim INRA Toulouse unit, where she conducts research focusing on the toxicology of mycotoxin. She's going to present us our work on Ergot alkaloid at those close to the EU regulatory limits. Viviane, is up to you.
Thank you, Marc. Thank you for the invitation. Today, is it okay for the presentation? Yep. So my presentation is entitled Intestinal and Hepatic Toxicity of Ergot alkaloids at doses close to the European Union limits. So, ergot alkaloids are mycotoxins mainly produced by Claviceps fungi. The fungi develop at various grasses and grains, forming a dark mass named sclerotia. So ergot alkaloids gained notoriety in the Middle Ages by mass poisonings in Europe, characterized by gangrene and convulsions, a condition known as St. Anthony's fire or ergotism. By now, more than 50 different ergot alkaloids have been identified. The toxicity is linked to their structural similarity with dopamine, noradrenaline, adrenaline, and serotonin, enabling the binding to the biogenic amine receptor and the interruption of neurotransmission. Although acute poisoning became rare, the ergot alkaloids are still a main source of concern due to the fact that they continue to be detected in cereals and cereal products in Europe and North America. For example, in this work of Maruo and colleagues, they observed the contamination of various cereal products in Belgium. Moreover, certain investigators highlighted an increase in the occurrence of ergosclerotia in the last few years. The regulations are based on the quantities of ergosclerotia. Codex Alimentarius fixed the maximum levels in wheat and durum wheat intended for human consumption at 0.5 and 5 grams per kilogram respectively. The European Commission fixed for animal feed the maximum content at 1 gram per kilogram of feed-containing cereals. Therefore, we are interested in investigating the effects of ingestion of ergot-contaminated feed at concentrations close to the regulatory limits. We mainly focused the research in pigs because they may be exposed to the ergot alkaloids. Philippe told us earlier about the exposition of pigs. And the pig is also a choice species as biomedical model for human health and disease. The intestine was a focus on our research because it's the first site of absorption of toxins, and also it represents a protection barrier preventing the entry of antigens. And this barrier, this epithelial integrity, is in fact made of various parts like tight junctions, proteins, mucus, and cytokines. Also, we focused on liver because metabolization process of xenobiotics, including mycotoxins, are made mainly at the liver. So for the study, we used 72 pigs of 30-40 days old, 12 males and 12 females per group, and they are assigned to 3 groups. One group that received 0 grams per kilo the ergot, one group received 1.2 grams ergot per kilogram, which is a dose close to the limit fixed by the European Union, and a third group that received a higher ergot dose of 2.5 grams of ergot per kilogram. They were exposed to the diets for 28 days, and the weight and feed consumption were registered at the first 14 and 28 days of experiment. At the end of the experiment, 6 males per group were euthanized and necropsied, and the liver and the intestine was sampled for histological analysis, and in the case of the genome, also for gene expression analysis. During the exposure period, no typical symptoms of acute toxicity, such as convulsions or necrosis, were observed in the animals. The daily feed intake of the animals consuming the higher dose were decreased from the first 14 days for the group with the higher dose, and for the group receiving the dose close to the regulatory limit, this reduction in the feed intake was seen at the end of the exposure period. During all the exposure period, the group receiving the higher dose of Ergot showed about 18% of reduction in the daily feed intake. Also, this reduction in feed intake led to a decrease of animal weight gain that was significant to the group that received the higher dose of Ergot. In the liver, the main histological findings included disorganization of hepatic cords, inflammation, and vacuolation of hepatocytes, megalocytosis, and necrosis. And furthermore, animals fed contaminated diets, they presented a significant increase in lesional score. Exposure to Ergot led to mild to moderate lesions of the jejunum of the pigs. The main histological changes observed were villi atrophy, edema of lamina propria, and cytoplasmatic vacuolation of the enterocytes. And as indicated for the lesional score, we observed an augmentation of this lesional score for the group receiving the higher dose of Ergot. The villi high was decreased at both groups receiving Ergot, and for goblet cells that are responsible for the production of mucus, we observed a decrease in the number of the goblet cells. So, as we found interesting differences between the groups at the histological level at the jejunum, we made a QPCR study of the analysis of the expression of levels of some of 34 genes that coded for junctional proteins, inflammatory and immunological mediators. For most of the genes involved in maintaining the structure of the intestinal mucosa, and overexpression was detected. So The study revealed that the ingestion of ergoalkaloids at doses close to the regulatory limit induced a reduction in the performance and morphological and transcriptomic changes, suggesting that the regulatory limits is not adequately protective. I would like to thank CNPq Brazil for the postdoctoral fellowship that I received, and all the colleagues that participated in this study, and the departments. Thank you, and if you have questions.
Thanks a lot, Viviane, for this nice presentation. So for the moment, I don't see any questions, so I may start myself. About the liver damage that you see in histology study, did you confirm them by doing some blood sampling and measurement of blood parameter of liver damages?
Yes, we did the analysis, biochemical analysis. Mm-hmm. And but we could not put the data by now because we have little time. We have some parameters that were different, but they were not classic markers for hepatic toxicosis.
Okay, thanks a lot. And what's your feeling about this? So I see the data, and that you clearly decrease the number of goblet cells in the gut, like half of them, 50% inhibition. But in the meantime, you have a massive increase in the MYC2 and MYC1 expression. MYC1 can be different because it's not associated to goblet cells, but MYC2 is really dependent of goblet cells expression. So how you can explain this? So reducing the number of goblet cells, but in the meantime increasing the mRNA coding for MYC2.
Well, we can understand this as attempting of the tissue to recover the fact that there are less goblet cells at the tissue.
By any chance, did you measure the mucus production itself? Not the mRNA, but really the presence of mucus in the gut?
Sorry, I didn't understand.
I mean, as your hypothesis is to say that the decrease in goblet cell is the gut try to compensate this decrease by increasing the expression of the muc.
Yes, yes.
Have you tried to measure the amount of mucus, the gel itself, really present in the gut of the exposed animal? By immunostaining or histology, or even, I mean.
I don't. The part of the histology is a colleague of us, of University of London, that makes the histological part, and usually they have this special markers to effectively quantificate. But I don't know. I think that for this study, it was not made.
Viviane, except if someone else wants to ask something, I have a last question that is wrote by some of the participants, because after we will be short in time. There is two questions. One of the questions, so Laura, it's not Laura Soler. So two questions. So I hope we will be able to answer them quickly. So Maria Elena de Lima is asking, are the alkaloids able to cross the brain-blood barrier, and to affect the brain? And I'm sure that yes.
Yes.
And the second question is from Laura Gamiz Gracia. Sorry if I spell it wrong. Did you measure the concentration of ergo alkaloid in the feed or only the
Yes.
concentration of the sclerotia, sorry.
Yes, the quantification of ergo-alkaloids, it was made by, and we quantified, and it was about 1 microgram per kilo.
And about the blood-brain barrier, even if I am
Yes.
for myself, but
Yes.
I'll let you
Yes.
confirm it. So I hope we answer all questions. And now I will give the microphone to Isabelle for the last speaker of today.
Okay, so last but not least for today, we have the last presentation from Laura Soler. Laura studied veterinary medicine and graduated from the University of Murcia in Spain. She did a first postdoc in the Catholic University in Leuven in Belgium, and then a second one at INRA Val de Loire in France. They were both focused on pig intestinal pathophysiology and clinical proteomics. In 2017, she joined INRA Toulouse, Toxalim, as a junior research scientist within the team Biosynthesis and Toxicity of Mycotoxin, and she's interested in applying system biology approaches to reveal the toxicity of mycotoxin. Laura, the floor is yours.
Thank you very much. So I'm going to finish the webinar today, and I'm going to still speaking about mycotoxins. So I'm going to speak about aflatoxin B1, which is one of the main mycotoxins known, and its precursor versicolin A. So I would like to start by mentioning that most of the things I'm going to show here are not mine. They have been directed mainly by our colleague Olivier Pouel, and they've been performed by mainly Thierry Gauthier, Karina Yubi, and Clémence Boudin. So I acknowledge their work on this. So just to start, aflatoxin B1 is a mycotoxin that is produced by moles from the Aspergillus genera, mainly Phlebus and Parasiticus, and contaminates mainly maize, rice, and nuts, traditionally in tropical and subtropical areas. But now with global warming, we're starting to see more frequently contamination of aflatoxin south of Europe. So it's getting here. And aflatoxins, and more specifically aflatoxin B1, are known to be hepatotoxins that show a very high carcinogenicity in humans, inducing mainly liver cancer. So this ability to induce cancer is performed by a derivative of aflatoxin B1, if we focus on aflatoxin B1, which is produced in vivo because the molecule needs to be bioactivated by certain cytochrome P450 enzymes into a very reactive epoxide form that will make DNA adducts, and this is the main way of the toxicity of aflatoxin B1. The aflatoxin B1 during the synthesis has many intermediate precursors, and if we look at the structure of aflatoxin B1, the first precursor showing this special feature that allows the production of the epoxide derivative is versicolin A. So this metabolite versicolin A can accumulate significantly before inducing an increase in the level of aflatoxin B1, so before inducing last steps of the biosynthesis pathway. And we know that the conversion rate is not complete. So we think that actually, in theory, versicolin A should be accumulated together with the production of aflatoxin B1 in those species that produce aflatoxin, but also in other species that do not produce aflatoxin but stop at the stereomatocysteine step. And so we believe that probably versicolin A can be present in different food commodities. We don't have a lot of information on this. The occurrence of versicolorine A is not very well researched, but there are a few surveys, and we know that it is indeed present. Sometimes, even in samples do not contain a lot of aflatoxin B1, and sometimes it can exceed levels that might be, if you take aflatoxin B1 as a reference, could be really high. And we guess that it might be toxic because it does share this structure that may give place to an epoxide derivative, and also because it has another feature that has been linked with an ability to disturb mitochondrial respiration, and most specifically being an uncoupler. So in the lab, we were interested in studying this toxin, and we started by checking what is the cytotoxicity in different cell lines. We observed that versicolorine A is more toxic than, more cytotoxic than aflatoxin B1 in most cell lines we studied. So if we look at the pink lines, you can see that for liver cells, this is the case. Also for LS174T cells, which are intestinal cells, we can see a higher cytotoxicity level, which is also seen in other intestinal cell lines, such as CACO2 cells and IPEC1 cells. And regarding the cytotoxicity effects, one of the things that were more striking is that if we look at the ACHN cell line, which is a kidney cell line that does not possess cytochrome P450, active cytochrome P450, cytotoxicity is as big as in any other cell line. So it does suggest that versicolorine A doesn't need to be bioactivated to be cytotoxic. Next, we were interested in looking at genotoxicity. Here we also saw some changes regarding the different cell lines. So if we look at the genotoxicity measure as by the phosphorylation of histone H2HX, which is an indirect marker of DNA damage, we see that aflatoxin B1 is more toxic for liver cells, FG2 liver cells, which was later confirmed. And this is independent of P53 transactivation, because both toxins were able to induce P53 in a similar way in the same cell line. But what was quite relevant is that both toxins were either likely genotoxic for IPEC1 cells or much more genotoxic, for instance, for CACO2 cells. So if we look at the phosphorylation levels of the histone H2AX, or DNA damage as measured by COMET assay, or the induction of micronuclei, we can see that indeed versicolorine A is very much toxic to intestinal cells, genotoxic. So in general, all this data, what suggests is that depending on the metabolic ability of the different cell lines, we had different results. So what it suggests is that we know that for aflatoxin B1, toxicity depends on its bioactivation, but it does not look to be the case for versicolorine A, at least not in full. And it looks that from all the cell lines that we observe, it looks like versicolorine A is especially bad for intestinal cells, and it does not look to have the same mechanism of action as aflatoxin B1. Indeed, some of these studies showed that versicolorine A is able to induce more apoptosis, is going to inhibit proliferation at lower doses, and it really increases oxidative stress in a very more intense way. And even if both toxins were able to induce the P53 response, even versicolorine A is able also to induce the same level of toxicity in cell lines that are that do not have an activity of p53, so it's independent of the p53 status. So even if it is still possible that the molecule could form adducts due to the bioactivation by cytochromes, it looks like replication stress may play an essential role in its genotoxicity. So to conclude, we can say that versicolorine A is a highly toxic molecule, and it probably doesn't share the same toxicity characteristics that its daughter molecule aflatoxin B1. It's particularly toxic to intestinal cells, where it can reshape the gene expression profiles, inducing a phenotype that is the typical one of cancer cells. And we also shown that versicolorine A is able to induce the transactivation of AHR and to induce the signaling of AHR, so increasing the ability of the cells to express some cytochromes, thus enhancing the genotoxicity of aflatoxin B1 by transforming it into these epoxides. So we need more research to describe all this mechanism of toxicity, and also to investigate what is exactly the interaction between aflatoxin B1, versicolorine A, and probably other precursors in the chain of synthesis of aflatoxin B1. So thank you very much, and feel free also to contact me by email if you have any questions.
Thanks a lot, Laura. I don't know if someone will have a question or not, but as we are short in time, I will start anyway, and then we will see. First of all, I would like to know if anyone or yourself have looked at the DNA adduct with the VRA molecule, because if you think that it could be genotoxic like aflatoxin, one expects its ability to bind with a covalent bound to DNA like aflatoxin, creating adducts. So is any data about that?
We are trying to see, we have started a study on the metabolization of versicolorine A. The main problem is that this toxin is not commercial, so we purify it ourselves in the lab. That means that for scaling up the experiment is quite difficult. So for the moment, looking at the adducts is a little bit difficult. We have started looking at what happens if we incubate versicolorine A with microsomes, and if there are some molecules that could look like an epoxide. And we are having nice results regarding that.
Good. The second question and the last, except if someone came up with this, another question. I was wondering if you use a good idea to use a microsome system. I was wondering if you ever try to see if VRA can compete with aflatoxin in a microsome system, meaning that if VRA can take the place of aflatoxin for the transformation by cytochrome in a microsome system without gene expression, with enzyme to compete as a substrate.
No, we didn't do that. Might be interesting to do that. Anyway, it looks like they don't have the same kinetics. From in vitro results, they do not have to have the same kinetics, and genotoxicity does not arrive at the same moment. So it might be, it might be not.
Okay, thanks a lot. Siska, I have a question. So please, Siska.
Yes, thank you, Marc. Hello, Laura. Congratulations, nice presentation on your work done. It's nice to see an inductor actually of a cytochrome P450 system. Usually mycotoxins are rather inhibiting agents, I guess. I'm thinking a little bit further. Now you look to the interaction afla with the versicolorine. But what about interaction with the metabolism of possible drugs that are substrates of CYP3A4? So many of them, a broad substrate specificity. Would that be interesting to study in future, to see that kind of interaction, the co-exposure?
Yeah, I think so.
Mm-hmm.
We started by aflatoxin B1 because it's the natural way. Like, we believe that they might be together.
Mm-hmm.
But of course, there is a high possibility that that we can see the same.
Yeah.
The toxicants.
And do you know other inducers?
Other inducers, yeah, but well, we know other inducers. They don't work at the same scale. Let's say like dioxins.
Yeah.
But yeah, there are other inducers that we are exposed to.
Okay, thanks. Good luck.
Elena, there is some question, but I think we can't.
Oh, we can get it.
Yeah.
There was a question,
If you have time, there's no problem. We can answer.
Okay.
Occurrence of versicolin A in food compared to aflatoxin.
Yeah, we don't really know that because most of the surveys do not include versicolin A. We do have some data that are shared by some nice colleagues, and it depends on the kind of food, and I guess it is probably biased by the thing that if we are looking for aflatoxin B1, then we will find a lot of aflatoxin B1 and probably less of versicolin A. In some samples, there is relatively a lot of versicolin A and less aflatoxin B1. But my humble opinion is that, if we are using samples that we are suspicious of aflatoxin B1 concentration, contamination, probably we will have less of versicolin A. But if we sometimes it will be contaminated by the same fungi, and then aflatoxin B1 is still not there, but we have high levels of versicolin A.
And there is a very last question, and then we finish, Laura. Could versicolin A be analyzed? Is there an easy ELISA to measure versicolin A?
That would be great. But no.
Maybe by competition with aflatoxin.
Yes, but I'm not sure that it has really the same mechanism of action. So it can be a measure, but I'm not sure how much it will be, up to what point it will be sensitive.
Thank you.
Thanks a lot, Laura.
Thanks, all of you.
Thanks to all speakers, chairs, and all participants of today's session. Maybe, Helena, you want to have the last words to announce the session of tomorrow? I don't know.
Yes, so thank you. Before we go, I would like to thank our chairs and our speakers of today. And yes, we have another live session tomorrow in the afternoon. So please be sure to sign and register for free in the conference website. I have shared with you the link in the chat. And well, that is all for now. See you soon. Hope to meet you in future events and in our talks tomorrow, well, in our webinars tomorrow and the next week. And I hope you enjoyed. Thank you very much for joining us. Bye-bye.
Thanks.
Bye.
Bye.
Bye. Thank you.